All right, welcome everyone to today's webinar on advances in adult Refsum disease research. This webinar will provide an update on key research initiatives related to adult Refsum disease. I'm Christy DeMarco, and I'm the president and founder of Global DARE Foundation, and I will be your host today. We are honored to have Wedad Fallatah, Dr. Wedad Fallatah and Dr. Rachel Huckfeldt as our presenters today. Dr. Fallatah will present first and discuss the ongoing Refsum mouse model study at Kennedy Krieger Institute, which is generating valuable pilot data for future large-scale research grants. Dr. Fallatah is a physician scientist with a deep focus on peroxisomal disorders. She earned her PhD in human genetics from McGill University, where she contributed significantly to the understanding of RCDP through natural history studies and preclinical testing of novel therapies. Dr. Fallatah is currently a postdoctoral fellow at Kennedy Krieger Institute and Johns Hopkins University, where she continues her research in peroxisomal disorders. Her work on the Refsum mouse model was recently recognized with the Best Poster Award in the category of biomedical research at the 10th Annual Johns Hopkins Postdoctoral Conference. After Dr. Fallatah's presentation, Dr. Rachel Huckfeldt will cover gene-agnostic clinical trials targeting retinitis pigmentosa, a condition affecting all individuals with adult Refsum disease. Dr. Huckfeldt is an Assistant Professor of Ophthalmology at Harvard Medical School and a clinician-scientist at Massachusetts Eye and Ear. She specializes in inherited retinal disorders with a particular focus on clinical trial of investigational genetic therapies. Dr. Huckfeldt completed her MD and PhD at Washington University at St. Louis, where she researched retinal development. Her postdoctoral work at the University of Pennsylvania focused on novel therapeutics, followed by clinical fellowships in medical retinal and inherited retinal disorders. She is the co-chair of the Foundation Fighting Blindness Clinical Consortium and guides multicenter clinical studies. Both Dr. Fallatah and Dr. Huckfeldt are also part of Global DARE Foundation's Medical and Scientific Advisory Board. Now on to Global DARE Foundation's mission. Global DARE Foundation was established exactly 5 years ago. We have our anniversary this month. We started in October of 2019. DARE stands for Defeat Adult Refsum Everywhere. Our mission is to promote worldwide awareness and better quality of life for all that are diagnosed with adult Refsum disease. Our goal is to support research, education initiatives, awareness campaigns, and advocacy. Together with our Medical and Scientific Advisory Board, driving research is at the center of what we do because we dare to believe there is a cure for adult Refsum disease. Now on to some housekeeping details. All participants are in listen-only mode without video. You can type your questions in the Q&A box at any point in the presentation. So we really highly encourage your active questions throughout the session, and those will be queued up for me to moderate after the session is over. There won't be the opportunity to unmute and ask your questions verbally, so I would highly encourage you to put your questions in the Q&A box. Today's session is being recorded for later viewing on the Global DARE Foundation's YouTube channel and website. Now I'll stop sharing and turn over the presentation to Dr. Fallatah.
Okay, let me share my slide. Okay, so can you see my slide?
Yes.
You guys? Okay, so Christy, thank you so much for giving me this opportunity and the Global DARE Foundation to present some of the exciting research actually from the Kennedy Krieger Institute. So my name is Wedad Fallatah, and I will present, we have a lot of ongoing research on ARD, but I will focus my talk for the novel adult Refsum disease mouse model study and updates on that. So this is just to give you guys a quick background about the study. So on the left side of the slide, you will see the metabolism of phthalic acid. And as you might already know, that there is a defect or deficiency of one of the enzymes, and that will lead to the accumulation of phthalic acid over time. What we tried to mimic in our research institute, so we developed a mouse model with a collaboration from Johns Hopkins University researcher, in which we also genetically modified this mouse to mimic what the genetic mutations represent in patients. And the goal for this is to help us to understand. So we'll have a model to understand the disease pathophysiology, how is the phthalic acid accumulation will affect the tissues, and how is that will affect lead to the pathology. And we also, this mouse model will help us to test future potential therapies. So the main aim or the goal for our study is to investigate and characterize not only the biochemical, but also the pathological and the functional impact of accumulation of phthalic acid. I forgot to say, so PA in my talk is related to phthalic acid. And the characterization, will be in the newly generated knockout mouse model that was fed with varying degree of phthalic supplemented diet. So I'll go ahead and show you the design for our study. So we have different mouse here, so mouse group, I would say. So on the left side, if you see my, let me see if you see my pointer. So here we have the control group, in which we have a mouse, a normal mouse. This is a normal control, in which the phthalic acid metabolism is normal because they have the gene. And the gray and the other colors mouse, those are the mutant or the ARD mouse model, in which we genetically modified them to mimic what we've seen in the ARD population. So we have about 4 groups in addition to the normal healthy control. So we have the mouse, the ARD mouse on a standard diet. So you will see the standard diet, and the normal control also on a standard diet, in which we didn't add anything or supplemented the food with phthalic acid. And then we have other 3 groups in which we supplemented the food that they are eating with different concentrations or variable concentration of phthalic acids. So we have 0.01%. This is the blue mouse, and will be the colors throughout my presentation. So all the results with the blue color will represent the 0.01% phthalic acid supplemented diet mouse model, and then we have a 0.03% phthalic acid supplemented diet given to the green colored mouse, mutant mouse, and then we have also the highest degree, which is a 0.06% phthalic acid for the red color mouse model. And then, so we have all the groups. We have a timeline. So we started at week 0 and we finished our study by 7 weeks. And then we also have a measurement. So what we did, so we give them the food by the starting of the study, and then we finished by the week 7. And we collected the blood sample plus tissues at the end. So we have 3 measurements of blood samples at week 0 before they are starting the diet, and then at week 3, which is like a midpoint of our study. And then at the end point, we have a blood, and then we have also tissue samples. And the goal is to measure the phthalic acid and to see if this mouse model mimic the gradual accumulation we see in a human patient. And then in the bottom, you will see, so we have different kind of tissue. The one in the purple box is subregions of brain. So we have the whole brain, but we also have a subregions collected differently just to have like a more accurate data about how the phytanic acid accumulated in these subregions of the brain, in addition to the spinal cord, eye, optic nerve, liver, and fat tissues. Okay, so moving on to the study method. So we have the diet. We supplemented with different concentration, as I told you, with the phytanic acid. And then we have at the end of the study, we have blood and we have tissues. We measured the phytanic acids in these samples by using a method called gas, or by using a machine, the gas chromatography mass spectrometry that's already set up by Moser lab in the Paroxysmal Disease Laboratory at the Kennedy Krieger. And this is a very like a quantifiable method for us to see whether we have an accumulation or not for the chow diet that we supplemented, and also for the mouse model that we generated. So this is the first part of our results. So when we prepare, Dr. Paul Watkins, he's originally started preparing the food for the mouse, was supplemented with the phytanic acid, and then I start helping him at the beginning, and then I start moving forward and generate the food for the mice. And for those, so I took a little bit samples from each of the diet that we prepared, and then we took that, measured the phytanic acid level in that diet, just to show that we have the diet, or just to confirm our result that we have a diet actually supplemented with phytanic acid. And you can see in the bar graph, there is a gradual accumulation in the expected accumulation, similar to the concentration that we've seen compared to the standard diet that we give to the mice. Then moving forward to blood. So today I will present the blood samples, and then maybe in the next talk we will talk about the tissues. So here we have the slide in which we have the blood, not only the blood, so we have the separated compartment of the blood. So we have the red blood cells and the plasma. And what we did, as I told you in the study design, so before we started the diet, we measured the phytanic acid level, and that's in the y-axis for these graphs. And then on the x-axis, you will see the different mouse models, so the blood samples from different mouse models depends on the concentrations. I will, in the next slide, I will show you the result for the 0.06, which is the highest concentration. But when we looked at the blood or at the plasma by the baseline, we don't see any differences in the group, which is as expected. However, by week 3, we see a gradual increase for the phytanic acid accumulation in the blood samples. And this is actually significant compared to the mutant mice on standard diet. And then this is actually increased more, or gradually accumulated more by the end of the study, or the week 7 end point of the study. When we looked at the 0.06%, we also see that here we collected the whole blood just to give an idea if we have the same accumulation, whether it's in the plasma, it's in red blood cells, or in a whole blood. So at the baseline, we didn't see any differences between the groups. And then over time, we see this accumulation, which is the highest by the endpoint of the study. Here, I'm just giving you the data in a table in which we have the numbers of the amount of phytanic acid as a mean of the group or an average, and then the standard deviations. And this is just to give an idea how it differ. Even for week 0, we can see there is a little bit of the phytanic acid, but after the supplementation, this amount of phytanic acid is significantly increased in the blood of those mouse groups. So to summarize, so we started by studying the biochemical phenotype. Mainly, we measured the phytanic acid level in a blood sample of this is newly generated ARD mouse model, and we were able to mimic what we see as a gradual accumulation of phytanic acid level in blood samples. And this level, I hope I showed you that these levels are actually correlated with the phytanic acid concentrations that we supplemented in the diet. We've seen that the phytanic acid mice, the mutant mice on 0.01% and 0.03%, they showed about 4.5 to 22.4 increase in phytanic acid level. And we showed that the 0.06% showed the highest endpoint of phytanic acid accumulation compared to the other concentrations, with a 26.6-fold increase in phytanic acid. So our future step, or the next step, actually this is an ongoing research. So the work that I present was only for the last year, but for this year, we are focused on evaluating phytanic acid level in tissues, all the tissues, and we are now in the step of processing the tissue and analyzing the data. As I can tell from the tissue, as far as in the liver, we've seen a little bit of the accumulation also of phytanic acid in the tissue. For future studies, we are hoping to evaluate phytanic acid accumulation, not only by the biochemical data, but also we will try to see the impact on the tissue pathology by directly looking at the tissue, and also do some functional analysis, especially for the brain and eye. And our results confirm that the ARD mouse model accumulate. So that's mimic what we've seen in the patient, and they are like perfect model for future therapeutic intervention. With that said, I would like to thank the Global DARE Foundation for their help for funding these studies. I would like to thank the KKI research. This is a huge effort from the whole team. I'm only part of that team, and it's actually a great team here at the KKI, and also our collaborator at the Johns Hopkins University. I would like to thank my funding for the fellowship, the foundations that helped to fund the position, and I will be happy to take all the questions for you. Thank you.
Thanks, Dr. Fallatah. And now we'll actually hold the questions until the end and have Dr. Huckfeldt do her presentation.
Thank you, Christy. Can you confirm that you can see my slides?
Yes, I can.
Perfect. Okay. Well, thank you for the invitation to speak about a different aspect of research for retinitis pigmentosa and RP in the setting of Refsum. It's really a pleasure to talk about what is happening in clinical trials and what I hope more of will happen. I do have disclosures that are related to being a investigator at Mass Eye and Ear for clinical trials, and also helping companies that are designing clinical trials. What I want to start with today is just a reminder of what all goes in. And I'm sorry, my slideshow is advancing on its own. If you can bear with me a second, I'm going to see if there's a timer set, because that's going to get really annoying. Okay, we'll see if this helps. Okay, what I want to talk about are first quickly just the stages of clinical trials. Second, some of the general strategies and current examples of those strategies being tested in trials. And much of that I'm going to focus on gene-independent or gene-agnostic approaches. And then I'm going to end with my little soapbox about what I think is needed to further clinical trials for any of the retinal dystrophies like RP that I see in my clinic, including Refsum disease. So just as a reminder of all of the work that goes into bringing a investigational therapy to people and patients, it really starts with the kind of rigorous, robust lab science you just heard about in the prior talk. And my colleagues at Mass Eye and Ear, we really take this part seriously when we are thinking about joining clinical trials. We want to make sure that a possible therapy has really been put to the test rigorously in whatever models are available before it comes to clinic. I hate the phrase, guinea pig with respect to people. Any therapy really does need, there are questions that can't be answered in these preclinical models, but this part is so critical in terms of making me feel confident that I'm not making a bad choice by bringing something to patients. And then we go to the core phases in people of clinical trials. So starting with stages like phase 1, really to just make sure that something is safe in people. In phase 2, starting to look more at efficacy. Does something even work, while also paying attention to side effects? And then in pivotal phase 3 trials, really focusing attention on verifying that something is efficacious in larger groups of people. And this little schematic was made for things like high blood pressure or diabetes, where there are huge numbers of people that can be enrolled in clinical trials. The picture is really a little different for retinitis pigmentosa and other retinal dystrophies, where there are smaller numbers of people available for any of these trials. And so in this setting, phase 1 and 2 are really combined together. So rather than phase 1 starting in unaffected healthy people, there's a phase 1/2 where the initial step of that is looking at safety. And then in the later parts of that initial effort, thinking about efficacy. And then I want to stress that even if something makes it all the way through phase 3, is taken to the FDA and approved, that is not the end of what we know about it. So sometimes there will be very formal post-approval monitoring. Other times, we just learn a lot more when we're using a new drug in real-world clinical practice. And I think probably many of you are aware that if you are interested in what is happening with clinical trials, the website clinicaltrials.gov is a good resource. And in this screenshot, I had searched for retinitis pigmentosa. I had put in some filters, I think at this point for active studies, and I got back a large number. I got 65 studies back. That can be brought down to a smaller number with careful use of filters. So in this case, I started with that 65. I specified I wanted a phase 3 trial, and obviously that will be interventional. And this screenshot is from a year ago. At that point, it just gave me one trial back. But clinicaltrials.gov is very dynamic. So it's a resource to kind of peer in on from time to time, just to see what is out there. It comes with a big caution, though, which I've highlighted in red here, that the US government does not review or approve the safety and science of all studies listed on this website. And there are certainly some listings for retinitis pigmentosa that are not things I would steer any of my patients towards. And what I've superimposed on this is the front page, or the front part of a article in the New England Journal of Medicine talking about a number of people whose vision was severely impacted to the point of blindness after receiving a so-called experimental treatment for age-related macular degeneration. So most of what you find on clinicaltrials.gov will be legitimate. Not all of it is. So just, bring that note of caution to it. But if we kind of step back now and just think about, what would we like to have to treat RP and other inherited retinal disorders? What I would like is to have a therapy that is ideal for somebody, regardless of what stage of RP they have. And let me say that better. Recognizing that different therapies might be better for different stages, we all want something, options for all of the stages. And I'm going to show you a little bit of a busy figure. This is taken from a paper by Dr. Jose Sahel and colleagues that really illustrates why we have this need for multiple types of therapies. So in this image in the top left, it shows a schematic of a retina in cross-section where cones are in orange, rods with the little pointy triangle on top are in green, and that layer of light-sensitive photoreceptors, rods and cones, connects to other layers of the retina shown in gray. And this first example that I'm indicating with my mouse in the upper left can be seen as an example either of a normal retina or a retina that has a genetic condition that hasn't caused any anatomic loss yet. The vision or anatomic or functional loss, so vision may still be normal. As we move down to the bottom left corner of the screen, we're transitioning to a phase of RP where the rod cells are less healthy, and I'm realizing I read my shapes wrong. So it's actually rods in orange and cones in green. I apologize for that. So in this lower left corner, we see fewer rods, even as the cones persist. And that's the stage where there are visual effects of our RP. As RP progresses, the cones become less healthy. So anatomically, they start to change, and that has its own effects on vision. And finally, when the cones are lost, that represents just a different challenge in terms of how we treat RP. So again, the goal is to have treatments to address all of these stages, recognizing that we're going to need different things, different interventions at different stages. So if we focus for a moment on these early stages with normal vision, ranging to loss of rods, these are ideal moments at which to bring either gene-specific therapies or neuroprotective therapies, that is, therapies that just help the retina maintain its health, maintain its function, even in the setting of a genetic defect. And I'm going to spend a moment on gene-specific strategies, even though we don't yet have one for Refsum disease in the works, or in clinic, just to provide some context for some of the other types of therapies I'll talk about. So, a core tenet of molecular biology and gene therapies is what we call the central dogma, where the DNA that encodes our genes ultimately results in the proteins that are the core of everything our body does. But the DNA has an intermediary step. The genes are, the DNA is transcribed into RNA, and then the RNA is ultimately made into protein. And so this is important from a therapeutics perspective because it represents multiple points at which an intervention might be possible. And increasingly over even the last 2 or 3 years, we've seen just a new set of tools, a new and growing set of tools applied to targeting DNA and RNA. And I think that is really exciting and hopefully just means good things for anybody who is working on these treatments or needs these treatments. But what I'm going to focus on very briefly today is two specific DNA-based therapies. One is gene replacement or gene augmentation. And in 2017, the FDA approved the first genetic therapy for any single gene Mendelian condition. That was Luxturna, which was approved for a condition that's similar to RP in some ways, but manifests much, much earlier in life. And the basis of Luxturna is using a virus to deliver a full-length, correctly spelled copy of a gene to the retina. And by doing that, and it's kind of nicely cartooned in this diagram, the idea is to make normal protein from that normal gene and restore cell function. And Luxturna was very important proof of concept that this approach was feasible and could be successful. And there is a really nice webinar on the DARE website from a scientist from a few years ago going more into depth about this in the setting of Refsum disease in terms of preclinical work. In recent years, we've heard a lot in the media about genome editing, sometimes referred to as CRISPR. This is a different approach where rather than replacing an entire copy of the gene, it's like taking an eraser and a red pencil, or in this cartoon, some scissors, and just making a very specific correction. And there are situations in which this is a better approach than gene augmentation. It has the limitation, though, that your tools are very specific to a particular misspelling in a gene. So it's not quite as one size fits all as gene replacement. And again, I don't yet have these kinds of efforts, in clinical trials to tell you about for Refsum disease, but this is to just provide context for some of the other things I'll move on to. So taking a step back, there are dozens and dozens of genes that cause RP, and this process of gene replacement trials, genome editing trials, it is lengthy, and it's one gene at a time. And across, if you take 100 people with RP and you do genetic testing for all of them, you're not going to find the precise genetic answer in all of them. So having gene-independent or gene-agnostic therapies is important for multiple reasons. And so now moving into that for the rest of this part of my talk, we're going to talk about neuroprotection first, and then the idea of vision restoring therapies. So again, neuroprotection is the idea of delivering some kind of medication or intervention or drug that helps a retina with RP fare better, helps it repair or sort of counteract some of the pathways that lead to neurodegeneration in the setting of RP. And a neuroprotective approach might be oral. For example, there's an FDA-approved medication for Tylenol overdoses called N-acetylcysteine that showed promising results in a small pilot study. And this drug, which we abbreviate as NAC, is now being tested in a phase 3 trial that's being run by Hopkins. But this is an international trial happening at over 25 sites. And if it is successful in showing that NAC has therapeutic benefit for the retina and people with RP, this is something that, again, safe and effective may result in changes in our recommendations for people with RP. Neuroprotective drugs or approaches can also be executed at a genetic level. So we know from many years of lab research that rods make something called rod-derived cone viability factor. And in its absence, cones are kind of more susceptible to degeneration. So a company called Sparing Vision is conducting a first-in-human phase 1/2 trial of delivering the gene for rod-derived cone viability factor and a similar factor to the retina using the same kind of approach that we use for the gene therapy Luxturna. This trial is right now limited to people with RP caused from a very small number of genes for which animal models of the proof of concepts for this experimental therapy exist. But we are excited to see what comes of this. And there's another company that's an example of this called Ocugen that is modulating some of the nucleic receptor pathways in the cell by expressing NR2E3 in the retina. And they've gone from phase 1/2 to a broader phase 3 trial, broader in terms of who it enrolls. And then finally, a neuroprotective approach may actually be stem cell based. So we think about stem cells as rebuilding light restoration and replacing degenerative parts of the retina, but they have a potentially broader approach. The company JSight did trials a number of years ago, really focused on what the effect of neuroprotective or neurotrophic factors released by stem cells might be if a aggregate of stem cells was injected into the vitreous jelly of the eye. And they are moving into more advanced stage phase 3 trials, I think, in the near future. So again, just another way of trying to protect the overall health of the retina. But when we move into these later stages of RP, where there are no longer rods and where the cones are less healthy, then we start to think about should we take more of a vision restoring strategy? And this, again, can be by stem cells, can also be by means of an approach called optogenetics, which I'll touch on more in a moment, and also retinal prosthetics or chips. I'm not going to say much more about chips, but what I'll say is that there's multiple opportunities for what we learn about setting an experimental treatment in one retinal disease to help other retinal diseases. So there is some pretty cool retinal prosthetic research being done right now for age-related macular degeneration. And I think successes there may hopefully result in a looking at these therapies in the realm of retinal dystrophies and RP. I want to spend a moment on stem cell therapies, though. The premise with stem cells is that regardless of their source, we are trying to use them to make new rods and cones that can replace the missing rods and cones in a retina with RP or another retinal disease. And more, we have multiple options in terms of where we derive those stem cells from. And as one example of that, we can take a skin biopsy or even a blood sample from an adult. We can turn that cell into a stem cell, and then we can turn those stem cells into either very precise types of cells like rods and cones, or the retinal pigment epithelium that sits under rods and cones. Or we can make a little miniature retina in a dish. That's what this retinal organoid cartoon in the lower left-hand corner refers to. And these are actually multi-layer structures that have rods and cones as well as the inner layers of the retina. Regardless of what the precise output is of the stem cell approach, whether we're going just for rods and cones or for a multi-layer structure, those stem cells need to be put, or the stem cell product needs to be put in the right place in the retina. And if our goal is not neuroprotection, but restoring vision, that means we need to put stem cells between the retinal pigment epithelium. I'm circling that here. Those are kind of a support layer for the photoreceptors, and then between the inner layers of the retina. So in this cartoon, you see a unhealthy looking photoreceptor, and the goal is to put the correct layer, the correct type of stem cell there, whether it is rod and cone precursors or whether it is, some companies might be taking more of a sheet-like approach. And I think stem cells are an example of an area where we learn a little bit more every time we do a first-in-human clinical trial. And so we had a stem cell trial at Mass Eye Near now a number of years ago that I think taught us about scientifically and clinically what should be done differently. I think there are multiple companies working in this space right now that hopefully will be bringing first-in-human trials to clinic in the near future. So there are technical challenges, but there are very, very smart and insightful researchers and clinicians working on them. Going to move on to optogenetics. And optogenetics takes a different perspective. It says, let's not restore the parts of the retina that have been made unhealthy by a condition like RP. Let's just start the process of vision a little bit later in the retina. So optogenetics uses a viral vector like we use for genetic therapies to deliver a gene encoding a light-sensitive protein to other parts of the retina. That's shown here with the purple opsin channel. This is the light-sensitive channel. In this particular example, it's targeting the bipolar cells that live in the middle of the retina. Those that have received this opsin are purple. The idea is that when light strikes these cells, it elicits the electricity that is really the core of starting vision, and again, just starts that process of vision a step later. These cartoons highlight targeting bipolar cells and retinal ganglion cells with optogenetics. And I'm sorry, I said the purple meant the opsin. It's really the green indicates what the vector is targeting, whether bipolar cells or retinal ganglion cells. But there are also efforts to target so-called dormant cone cells in the retina as well with optogenetics. And many of the companies working on this approach are using a special kind of goggle to help amplify the signal that is really at the core of this process. There is also a company that is trying it without amplifying goggles. So I think this is in its pretty early days, but there have nonetheless been some really encouraging proof of concept data that have been released by a number of companies. So, you know, another stay tuned and reason for optimism. But I want to just come back a second to things that are true of all of these trials, whether they are gene specific or, you know, an oral neuroprotective agent or optogenetics. And that is that there is no such thing as a risk-free clinical trial. The risks are going to be you know, better defined in a phase 3 trial than they are in a phase 1/2 trial, just because by phase 3 we have more experience treating people, and we've, you know, learned more about what the risks might be. And a surgically delivered intervention is going to probably always have a little bit more risk than an oral intervention. And that's a part, you know, that's separate from what has the best efficacy for a given person with a given condition, or the best potential efficacy, I should say, because there's really no guarantee of benefit at any point in clinical trial stages or phases. What I want to also say that's not on this slide and is rarely contained in informed consents for clinical trials is that there is always the possibility that somebody will be in a clinical trial. Maybe whatever they received works or doesn't work. Let's say there's no risks that were damaging or had any complications for them, but there's always the risk or the possibility that receiving a experimental intervention may keep somebody from receiving other either investigational or approved therapies down the road. And that has to do with the response of the immune system to any viral-based therapy that's given to an eye. And in some cases, the surgery needed to deliver a therapy may mean that it's harder to go back to that same eye with another intervention in the future that also requires a surgery. So something that I always try to talk about when I'm consenting somebody for a clinical trial. I think it's also it's important not to minimize what we ask of people who are in clinical trials. We get to know each other really well, especially in a phase 1/2 study. There's typically a long list of assessments that are done at each visit as we try to understand a therapy, an investigational therapy as well as possible. For any gene therapy study, studies are 5 years long at the guidance of regulators. And by the time something gets to phase 3, there may be fewer visits, there may be fewer assessments, but we still see each other frequently. So just another reality of clinical trials to keep in mind. So I'm going to shift direction in the next few moments and just pose this question, you know, what else do we need if we want to be sure a therapy is working or understand its effects on a group of clinical trial participants? And I think one very important answer to this question is we need to understand how a condition just develops and progresses without intervention. And we often know that over long periods of time. But we need to know it on a time frame that is relevant for clinical trials. So rather than saying, this is how things look at 5 years versus 10 years, what changes over 6 months or 2 years? So that's a different time frame. Thinking about any intervention for RP, for that particular either stage of RP or genetic form of RP, we want to understand how different aspects of retinal function and visual function, as well as different aspects of retinal structure change. And we want to understand what the relationships of those retinal features are to other variables, whether it's to age, other health issues, the particular genetic misspelling. And it's pretty easy to think about, in the setting of metabolic conditions, how does what's happening with RP relate to the underlying, metabolic problem? And I want to quickly stress the efforts of a group called the Foundation Fighting Blindness Clinical Consortium. Christy mentions in her introduction that I am the co-chair for this group, and it is comprised of over 40 clinical sites in 15 countries. All of the sites are clinics that have expertise in patients with inherited retinal disorders like RP. And our mission is really to provide the data that will help accelerate the process of developing and testing therapies for RP. One of the big ways we do that is with natural history studies. We started out with a few gene-specific natural history studies, but I want to highlight one in particular. This one is led by Jose Sahel, and it's recognizing that going gene by gene, it will take us a long time to learn everything we want to know. So this study that we abbreviate as UniRare, the Universal Rare Gene Study, is really open to almost any genes that cause RP or other inherited retinal disorders. The PHYH gene is on the list of accepted genes. And this study will ultimately have 1,500 participants across our different clinical trial sites. Most of those participants will come in for a single visit to collect standardized data on them that we can then compare to standardized data on all the other individuals and genes for which we get data in this. And the goal is that it will help us kind of learn cross-sectionally and across genetic forms of RP and other inherited retinal disorders. So I've highlighted the sites in the US in this map, and there's also a link to clinicaltrials.gov. So that was a little bit of a whirlwind. I will kind of end by saying I'm really optimistic about many of the things that I got to tell you about today, and hopeful about what's to come. Thank you.
Thank you, Rachel, and Dr. Fallatah for your wonderful presentations. And Dr. Fallatah, if you want to come back on the screen, we're going to do some Q&A. I don't have anything in the Q&A box yet, but I do have a couple of questions that I can while folks are thinking of their questions or trying to get them into the Q&A box. So I'll start out with one for you, Dr. Fallatah. Why do you think the pilot studies in mice are so important for future therapies? And maybe Dr. Huckfeldt, you might have some thoughts on this as well based upon your presentation. But why is it so important to do these pilot studies in mice?
Thank you, Christy. That's a very important question that we all get all the time. As you can tell, so we don't know a lot about this disease. And in order to know about this disease, sometimes you need some accessibility, so tissue and bloods. And this is sometimes it will be hard for patients. So that's first things to do in a model. So that will give you a better understanding and insight about the disease pathophysiology. And then another things, that is a lacking of a biomarker. So if you have a therapy that you want to test, or let's say you have a drug that you want to test, you need something to measure. So And then you need also a model to start with and to test that therapy before starting on patients, just for safety also. So the mouse model would be perfect for that if we would start with that. So if we have a biomarker, and then you can get the drug, you have a biomarker, and you can see a response if this is drug actually working or not. So these are maybe the two main things. I think Rachel, maybe she will have another also input. Yeah.
Yeah, I think mouse studies like this are so important. They help us understand how Refsum disease affects different organ systems, like she said, and really, I get super excited thinking about the potential of this model to understand retinal changes and what changes when, and then that is such an important foundation as for testing any investigational therapy. It is such a boon and such a good thing when there is a robust, well-characterized animal model. We don't have that for all of the conditions that we have clinical trials for, and it's just feels like a luxury when we do.
Yeah, we were very excited to take the great research that was done in the past and be able to kind of pick that up and start it over, and I'm excited, that the team at Kennedy Krieger was willing to do that. So thank you both for the answer there. We do have one question in the box before I go towards any other questions I have. So the question was, when I was diagnosed with RP at 30 years ago, I was told that there would be a cure in 20 years. Clearly, we're not there yet. In your opinion, what has been the biggest hurdles in finding a cure?
Yeah, thank you for that question. And that experience, which is shared by so many, is why I never put a timeline on things, because I think we are all hopeful and optimistic, but then it takes time. And so um, I think one of the hurdles has just been realizing how hard it is to take a promising scientific finding or idea and translate it into something that really works and works well enough to justify like any risk that comes with it. I think what we've seen for gene-specific therapies is an acceleration, fortunately, of some aspects of this process. So it took Luxturna like at least 20 years to go from first clinical proof of concept to FDA approval. I think we are hopefully in the years to come close to seeing a much shorter timeline. So some of this is figuring out the route that works, the pathway that works, and then applying that to other diseases. As kind of what I think of as like version 2 or 3, or maybe we're at 4 or 5 of stem cells starts to come into first in human trials. I hope that same acceleration process happens there too. So I don't have the answer, but I think there's a number of pieces. I will say that I think regulatory agencies sometimes get a really bad rap. I don't see them as an obstacle. I see them as protecting, like really doing their utmost to protect patients, and hopefully continuing to work hand in hand with us to make sure that we're not delaying getting anything to people who need it.
Yeah, it's one of the reasons we really want to start to build a relationship with the regulatory bodies, right? Because we do want safe and sound clinical trials in the future for people with Refsum disease, but we also need for them to understand what Refsum disease is all about, so that they can understand what we're interested in accomplishing with adult Refsum disease. What are the clinical endpoints that are the most meaningful for patients and things of that nature. So I think it's a long road, but obviously we'll are obviously actively trying to move down that road.
Can I add a little bit? So also because Refsum disease is a rare disease, so it's hard in the rare disease world to to get in therapy. So one thing that you guys really good or excellent about it is to establish this foundation and found all the patient. So now we start to have a natural history about the disease, especially for the rare disease. It's hard to see to find patients sometimes. even for physician, they don't have this awareness. But this is how we started, like a baby steps. But you started with creating the awareness in the medical community, creating awareness in the research, building a network between research and physician. And this is how we all started after the foundation. So now we have a mouse model that we can test, and then we also have going with the clinical trial. I know it's a long road, but at least when I see the Refsum disease compared to the others, I'm very hopeful. This will be like going much more faster than others.
Yeah. I love that you said baby steps, because I'm always saying
Yeah.
baby steps are better than no steps, you know,
Yes.
and I think for quite a while, no steps were being made. And I think we're making some good progress now. It
Correct.
will still be a long road, but
No.
but at least it's some forward progress and moving forward. A question
Can
I
I,
have
Christy,
for, go ahead.
can I just add that I also
Yeah.
think it's really important and valuable to have patient perspectives shared with regulators at all steps. So something I've seen noticed just being more overt and vocal at recent meetings is patient voices and patients saying, and I don't want to put the words of one person in the mouths of everybody, but saying like, look, if you could keep me where I am or slow me down, that would be so meaningful to me. And I think that particular voice hadn't been heard as loud in the past. And it's good for regulators to hear that as they think about the kind of endpoints that they're willing to approve for clinical trials, especially, as Wedad said, in the setting of very rare diseases.
Yeah, and it's exactly why we want to kind of get out part of our goals for the future. And we'll talk about this on Saturday when we meet to celebrate our 5-year anniversary, is that we really want to build that relationship with the regulators, get the voice of the patient out in front of the regulators so that they understand, even, you know, that they know what Refsum disease is, you know, and that and what's important to us. So I think that would be a great step forward, as you mentioned. One last question I think I have for both of you as we end is, you know, how would you recommend that people with Refsum support research? Dr. Huckfeldt, do you want to go first?
Sure. So I love the registry and the organization of this group because it makes sure that patients know or individuals know what's going on, and participating in every single thing may not be the right fit for everybody. The Center of Excellence that Christy has put together in Boston, I think, is also a really powerful opportunity for people who, you know, hopefully at some point in the future when we have more research studies for Refsum, a way to participate those in one site. Right now, though, anyone who is interested in just making sure that Refsum is recognized in big cross-sectional databases like the UniRare study, you know, find a FFB clinical consortium site close to you, and see if you're not already a patient of one of those doctors, see if you can be seen there, and you know, ask about the UniRare study.
Great. Maybe we can actually share some information about that with our community, so they can know where they might go for that. So,
Yeah.
go ahead,
Yeah,
Dr.
so that's great. I will just add for that. Just be an advocate for your for the disease that you are in, to help to educate not only the community, even the physician that you are. Let them know about the foundation that you have, the networking, even the scientists together. So I think just being like organizing this awareness week just to talk about it and learn from each other, that's really helps to support the research forward. And also you're learning from towards what's we are going and what's the update, and this will help, and then raise the awareness, and I think we're moving forward, so very positive for that. So yeah, great question.
Great. Thank you both. Thank you thank you, Dr. Fallatah and Dr. Huckfeldt, for the very informative session today on research. It's exciting to know that we're advancing work in our Refsum mouse model, and it'll set the stage for more advanced research in the future. I'm also excited to see all the great research that's being done on clinical trials around RP. I think there's obviously some good progress being made on those fronts, which it directly could have a successful impact for people with Refsum disease as we move forward. We appreciate both of your continued advocacy and work with us at Global DARE Foundation as well. So being really valuable members of our medical and scientific advisory board. I'd also like to thank the participants for joining and providing your questions. And so that ends today's session. So thank you both, and thanks everyone.
Thank you.
All right.
Thank you,
Bye.
Christy.
Bye.
Bye.